Hey everybody, it's time for another video tutorial with Dr. Ramey. Today's tutorial is going to be on the preparation of smears and simple stains. Don't let that word simple fool you though. There's nothing simple about simple stains. Alright, so we've got two different types of organisms we're working with. Now I'm working with live organisms, so I'm going to make sure I've got my lab coat on as always, my safety glasses, and I'm going to put some latex gloves on. Okay. Notice they're nice and form-fitting, tight. Okay. I'm going to get my gloves on first. Got materials. I got my materials all around me here. I'm going to work with the slant of Staphylococcus epidermidis, and we're going to work with a broth culture of Bacillus Megaterium. Okay. So the first thing we're going to do, I'm looking for a grease pencil. I'm going to go over here. If I don't have my own grease pencil or marker, maybe a Sharpie, I'm going to go borrow one. Okay. And I'm going to set up my sterile field. I'm going to need a flame. So first thing I'm going to do is, after I get all materials all set up and ready, I'm going to get my flame on. Okay. You can see there's some stuff missing out of some of these community drawers. If that were to happen to you, you would need to let me know. Okay. Alright, so I'm going to be working with brand new clean slides. There is a way that these slides can be cleaned, but I'm not going to make that part of this tutorial. I'm going to assume that this is a brand new slide or a slide that I have cleaned before I start working with it. Alright, so one of the first things that you want to do is mark your slide. So that's what I wanted to get that grease pen for. And I usually like to make one end of it excuse me, usually like to mark off one end of it, so that'll kind of be the end you hold. and if you're going to use the slide for one organism, you can kind of put a circle in the middle. Or in this case, we're going to be working with two organisms, so I'm going to split the slide into two halves there, where one end of the slide is my holding end, and the other, and then I've got a line down the middle, and I'm going to put one organism on one side. Put that there. I'll put one organism on one side and the other organism on the other side. Okay. And you can even label it if you want. You can even I might put Epi here and MEG for Megaterium over here. You can probably see that now, right? Okay. Now that we got that all set up, okay, I've already set up my sterile field. I've already disinfected the bench, done all of those things. Got my materials here. I've got a water dropper. I've got the simple stain we're going to use today, which is methylene blue. I've got my organisms over here, one in a slant, one in a broth. I'll do the Epi first, right? And because it's in a slant, okay, if you take a look here, in a slant, that's a solid medium, right? So if you're going to take a sample from a solid medium like a slant or a plate, you need to first put a drop of water on your slide where you're going to put your specimens. Okay, so on the Epi side, I'm going to put just one. I'm going to try to hold my hand so you can see from that side too. Just one drop, one small drop. Notice I'm being very careful not to allow the dropper to make contact with the slide. Okay. That's so as not to contaminate the dropper and then hence the water bottle. Now I'm going to put my grease pencil over here out of my way. I'm going to put my slide box out of my way here. I'm going to make sure that you guys can see what's going on here. Move that out of the way too. I'm going to go into my community drawer and I'm going to find my inoculating loop. Okay, if it's bent, then I can straighten it out first. And of course, I'm going to properly sterilize my inoculating loop. Okay, so find that sweet spot there, and red hot. Make sure that my entire wire, the entire wire of the inoculating loop, is properly sterilized in the flame. Then I'm going to give it a few seconds to let it cool. Then I'm going to, using good proper aseptic technique, I'm going to remove the cap. A couple of passes through the flame. I'm going to go down and very carefully get some organism. Now you don't need to bulldoze the organism out. You just need to go down and make sure you get some bacteria. You can see some on the side there. Go down and get some bacteria off of the slant onto the loop. Two passes in the flame with the test tube, recap, and now I've got some bacteria here on the end of my loop, and I am going to stick that into the loop, into the water drop that I put on my plate already, and I'm going to swirl it around, spread it out into the size, a circle about the size of a dime. Okay, and if you've got enough bacteria, you might even see a slight haze in the water now. Okay, again, more is not better. More is not better, but you definitely need to have some bacteria. So better too much than none at all, right? Especially when you're first learning. Now, of course, I'm going to sterilize my loop. It's kind of wet, so it's, well, I don't know about this flame. It's not, there we go. That's better. Okay. That's what I wanted. All right. So at this point, I would proceed, if I was only going to make a smear of one bacterium, but I'm making two. So before I proceed to the next step, I'm going to add the bacterium to the other half of the slide first. So this is Bacillus Megaterium, or B Megaterium, from a broth culture. Whenever you're working with broth cultures, you want to make sure that you have mixed the cells in the broth first by gently tapping it several times with your finger, kind of like so. Okay. And since it's been a little while and I've been talking since I've inoculated since I've sterilized my loop, I'm going to sterilize my loop again. We'll wait several seconds, and I'm going to pull the cap off of my broth culture of B Megaterium. 2 passes through the flame at a 45-degree angle. While holding the test tube at a 45-degree angle, I'm going to go in, dip my loop in, pull it out, 2 passes through the flame, recap. Done with that for the moment. And because this was the broth culture, I'm just gonna spread that liquid that's on my loop around right there on the microscope slide. Okay, it may not be quite as much liquid, quite as much as there was for the other sample, because we use a drop of water. Okay, and then finally sterilize my loop. Don't need it anymore for the moment. Okay, so now I have spread out my samples, my bacterial samples, on the slide, but the slide is wet. Okay, so I need to air dry. You have to air dry. Now you think, well, wow, that's going to take forever. Well, not so much. Okay, because you can use hot air coming from the flame. But first, I need to find my slide holder, which is a very high-tech piece of equipment called a clothespin. Then I'm going to grab that end of the slide that I marked as the holding end with my with my test tube, or excuse me, my slide holder, my slide holding clothespin. And now there's a lot of warm air up here. You don't want to get too close to the flame, but I'm going to use that warm air up there to dry my slide. Okay, so I might speed this up a little bit, but it's going to take a little while to dry that slide. Now one side's already pretty much dry, okay, but the slide that has the water drop on it, it's going to take a little bit longer. And you are going to get a little bit impatient, and you're going to start to get it closer and closer to that flame, and you will mess up your cells if you do that. Okay, so even though it will speed up the drying process, if you put it closer to your flame, it is not good for the cells, and you might destroy the integrity of the cell walls by boiling them or making them too hot, and your stain will not work, especially when we move on to more complex stains like gram staining. Okay, so you want to resist the urge to hold it too close to the flame, okay, for this point, or at this point. And you can see that it is drying, and I'm just being patient, and I'm not putting it too close. I'm not lowering the elevation at all, and it's almost dry. Okay, so we're going to be patient, let it finish drying. But it has to be 100% completely dry before you proceed to the next step, which is heat fixing. Okay, so what you don't want is you don't want your cells to wash off during the staining process, right? So in order to make sure that they are stuck to the slide pretty much permanently, we're going to go through a process called heat fixing, okay, which also kills the cells. So you make sure that you're not working with your organisms on your slide are in fact no longer viable. They're no longer alive, and that they will stay on the slide. They will not wash off the slide during the staining process. Okay, well that looks real good. Okay, everything's good and dry. You can see a faint haze on one side, then a very distinct haze on the other, indicating that I may have gotten too many cells on that side, but I did that on purpose, okay, for this demonstration, so you could see the haze. All right, I won't know for sure if it's too many cells until I take a look at them in the microscope. Now you're ready for heat fixing. It's got to be 100% dry, 100% dry. You're ready for heat fixing now, and heat fixing is very quick and simple. You are going to pass the microscope slide 3 times through the flame, lengthways, and you're not going to linger in the flame, because you can literally, it'll heat up and break the slide if you do that. So we're going to go 1, 2, and then 3 times through the flame. Very simple. 1, 2, 3 times through the flame. I didn't do it again. Okay, lengthways through the flame. Right, now we have air dried, we have heat fixed, so we're in pretty good shape. Want to be careful because that slide might be hot now. Okay, going to move this stuff out of the way just a little bit, and now I'm actually ready to stain the cells. So I'm going to look in my community drawer, and hopefully there is a staining rack. Okay, it looks something like that. Then I'm going to lay over the sink. Okay, so at this point, I might add, once you've heat fixed, you are for the moment done with the flame. So you should definitely turn your flame off. Turn your flame off when you're done heat fixing. If you want to do more cells later, then it's very easy to turn the flame back on. I'm going to turn the camera a bit where you can see what I'm doing over here. Okay, so I am going to, when I'm sure that you always want to handle your slides when you've cleaned them, or if they're brand new from the side. Okay, hold them from the side if you're going to hold them with your fingers. That prevents messing up your specimens, and if you're not using gloves, it prevents getting oil or getting oil from your skin or getting the slide dirty. Okay, which can affect the the outcome of your stains, right? So I'm going to set my slide over there on the rack. Hopefully you can kind of see that in at least one of the views. And I'm going to use this bottle of methylene blue, which is our simple stain. Okay. And I'm going to make sure I thoroughly cover the cells with methylene blue. Usually 1 or 2, no more than 3 drops is usually enough. That smear is a little big, so I think it took 4 or 5 drops there on that one. Okay. And I'm gonna keep an eye on the clock for 30 seconds. Okay. 30 to 60 seconds for the simple stain methylene blue. Okay. So I'm gonna make sure it's a good, more like 45 to 60 seconds. I'm going to wait, and then I am going to I'm going to wash the excess stain off. I'm going to wash the excess stain off with water. Now I'm not going to use my little water dropper here, little bottle, okay, because I would quickly go through all that water. But I've got distilled water in a larger squirt bottle, okay, and I'm going to gently, I'm going to angle, try to do this so I can make sure that you can see it. Should have turned this around the other way. I'm going to angle it down with by holding it, and then I'm going to rinse off the stain. You don't want to use pressure, because you don't want to rinse those cells off by using too much pressure. So you're going to gently rinse them off. If you've got a lot of cells, then the stain might be extremely dark, like on the one side where I intentionally put way too many cells. Or it might be so faint that you might not even be able to see it, like on the other side. So you can see this one's too dark, this side's too dark where I put too many cells. This side, you pretty much can't see anything at all. It's a very, very faint haze there where the cells are. That's probably going to be a really good stain right there, probably. Okay, and then the last step before I'm ready to turn this so you can see what I'm doing, okay? Before I'm ready to use my microscope the last step is I'm going to put I'm going to get some paper towel. I'm going to lay the slide down on one side of the paper towel, and then I'm going to press down. I'm not going to scrub or rub or anything like that, okay, because then you would wipe the cells off. And then I'm going to move it to another section of the paper towel, a dry section, and I'm going to keep doing that till I don't see any moisture coming through on the paper towel, which usually takes 2 or 3 times, where I'm just going to gently press straight down, not going to rub or anything, because then you might rub your cells right off the slide. Okay, I can still see a little moisture, so I'm going to do it yet one more time, and voila, I have my bacterial smear, 2 of them, they've both been stained with methylene blue. Okay, so fairly simple and quick process. But now the final step, the whole reason we're staining the cells in the first place, is to get a better look at them, right? So I'm going to leave my cells right here for a moment, and I'm going to go get my microscope. Except now I'm done with my stain for the moment. I'm not working with live organisms anymore at the moment because they're over here, and the organisms that are on this slide are now heat fixed, and hence they're dead. They're not viable, right? So I've got even if these were pathogens, they would no longer pose a health risk to me or anyone else because they're dead. And I'm going to start working with my microscope. Never ever wear gloves when you are working with your microscope, right? The only time you should be wearing gloves is when you're working with live organisms or when you are working with stains. Okay, now that I'm done working with live organisms, now that I'm done with the staining process, I can now remove my gloves, and only then should I go and get my microscope. So let me go over here and grab my microscope, being sure that I am holding it properly as I carry it. I'm going to set it up here and plug it in. Okay, looks like whoever used this last did a pretty good job with it when they put it away. Looks pretty clean as far as I can tell just by glancing at it. Okay. Now the inoculating loop has been sterilized and it is cool, so I'm going to put it back in my community drawer. I can put some of these other things in the community drawer to kind of get them out of my way and tidy up. And I've got my microscope slide. I am going to need oil. Boy, this community drawer has been ravaged, it would appear. Oh boy, there's oil in here that is not in its proper little container here that prevents it from spilling. Okay, I've got my oil, got my slide. Let's start using my microscope. So first, what the heck, I'm going to look at the stain, the really dark stain that's probably got way too many cells in it. No cover slip required, right? No cover slip required. Okay, I can also remove my safety glasses once I start using my microscope, and I'm going to start the process of visualizing my cells once I make sure that my ocular lenses are synced up good. Alright, excellent. Excellent. Okay. I'm ready to swing up to the next power. Do some fine adjustments there. Oh yeah. Got some beautiful looking cells there, I can already see. Even though they are a bit clumpy because I got too many. Once I get to 400x, I might want to find an area, if possible, that's not quite as busy. In other words, it doesn't have quite as many clumps of cells. Okay. And once I find an area that I kind of like a little bit better, I'm going to swing up to my 1000x or 100x oil immersion lens after I add my oil. Come on, drop off there. Come on, there we go. Remember, you don't want to touch the tip of the oil dropper to the slide. Swing my oil immersion lens ever so carefully into the oil, and I'm going to very gently and carefully make some adjustments using only the fine focus knob. And oh wow, got some beautiful cells. I want a little bit more light, just a tad more light there, and just some classic blue coccyx cells. Makes sense since those are Staphylococcus, right? All right. I'm going to flip this around where you can kind of see what they look like. All right. Let's see if I can do this. Some of my students are so good at this. There they are. Beautiful. Nice coccyx, and their arrangement is staph. So you can see they're kind of okay, here we go. Okay. All right. Now let's take a look at the other cells, the Bacillus Megaterium. I wonder if you can guess what the morphology of those cells should be. Bacillus Megaterium? Okay. I might be able to swing all the way over to where they are without adding any more oil. Let's see if I can do that. Okay, so I'm gonna go over and see what I can see here. Okay. And these on this side, I am finding some nice looking Bacillus Megaterium cells. I'm not going to show, well, let's see. Might show you. Okay. But they are, you know, methylene blue stains the cells blue. And on this side, I intentionally added much fewer cells. So got to spend a little bit more time hunting around for the cells, perhaps. But you can get a much better look at individual cells when you don't add too many. Okay, so you find a good area. You know, you search around and you find an area where you can find a good, you know, an area where there's several cells that you can take a look at. And I found a pretty good area here. Okay, so these cells are more rod-shaped. Some are very clearly rod-shaped. Some are less obvious so. Okay, you've got to be careful because some even look a little coccyx, but you have to be very careful about that because some cells will fool you, and you have to don't rush to judgment. Take a look at more than just one cell when you are, if it's not clear. Let me flip this around and so you can take a look. Okay, let's see. There we go. So cells are very small. In this video, I'm not sure if it's going to be clear, especially not the way I'm jiggling things around. Okay. You know what? It's an iPhone. I did the best I could. I did the best I could, but you're going to see for yourself when you work on this in the lab. And actually, they both came out pretty good. Actually, a little bit better than I was kind of anticipating, because I intentionally put way too many cells on one side and intentionally put far fewer cells on the other, and actually they both came out pretty good, right? So as long as you have some cells and as long as you are capable of finding those cells, they'll probably, your staining will probably look good. Okay, sometimes it's difficult to find the cells, even when the stain is good. It just depends on how many cells there are. But that's it. That is the tutorial for not only how to create, your heat prep your smears, but also to stain those smears with a simple stain. So a simple stain is where you just use one type of stain and there's no counter stain. So other types of stains like gram stain, which we're going to be doing next very soon, is a two-step or a two-stain process. Once with a simple stain, then you've got to de-stain and then do a counter stain. So very exciting.